Hello, my name is Ellen Jones, and I am a student at Haines City International Baccalaureate East. The goal of my project was to examine genomes of dengue virus for a biological marker of severity. Dengue virus causes a disease which has 4 distinct serotypes. After infection with one serotype, one is immune to the disease from that serotype, but will experience the disease from other serotypes more severely. As this would confound the presence of dengue fever versus dengue hemorrhagic fever, I decided to block this variable. The disease caused by dengue virus can be dengue fever or dengue hemorrhagic fever. Dengue hemorrhagic fever is the more severe form and can be diagnosed with the observation of acute high fever, hemorrhagic manifestation, hepatomegaly, or an unusual enlargement of the liver, and shock. This project searches for markers in the genome that will reveal how severe an outbreak or cluster will be, which is useful for the planning and use of resources to assist in fighting the disease. The genomes I used for this project were found on the Virus and Pathogen Database and Analysis resource, which allows users to filter various factors about the disease or the patient, the immune status, as well as whether the patient was infected with dengue fever or dengue hemorrhagic fever are 2 factors this database can filter. Basic local alignment search tools, or BLASTs, find matches, mismatches, and gaps in alignments of genomes. There are many types of BLASTs, but for my project, I used the Needleman-Wunsch BLAST on the National Center for Biotechnology Information to create box plots and show the entire genome and its matches. I also used the BLAST software on the Virus and Pathogen Database and Analysis resource, which gave e-values, which are the number of alignments expected due to chance, and bit scores, how large a database would have to be to find matches or to find a match based on chance. This BLAST shows statistical significance of the results. I randomly selected a primary dengue fever genome and a primary dengue hemorrhagic fever genome. The Needleman-Wunsch blast showed a 98% similarity between the genomes. I randomly selected a different hemorrhagic fever genome to align with the dengue hemorrhagic fever genome. The Needleman-Wunsch blast returned a similarity of 55%. I then looked at each mismatch in the first blast between the dengue fever and dengue hemorrhagic fever genomes. I found how many times there was an alignment between the dengue hemorrhagic fever and the Algorma hemorrhagic fever I randomly selected, which yielded a similarity of 34.51%. Though there was not enough evidence to suggest an association between the genomic differences of the dengue fever and dengue hemorrhagic fever genomes, the e-values and bit scores show that the results found were not due to chance. We fail to reject the null hypothesis that there is no association in the genomic differences between dengue fever and dengue hemorrhagic fever when blocking for immune status. Further continuation of my project may provide enough evidence to reject the null hypothesis. If a marker is found, it could be observed through other hemorrhagic fevers to research impacts of the genome on severity and what makes the disease hemorrhagic.